Just in case, I'm gonna put this over here. So if anybody is watching online, which I know we have at least one person, but I didn't look at the numbers. Where we have 2 right now. So you guys can talk over your microphone if you want. This computer speakers don't really work. So probably through the chat is the best way if you guys have questions. Let me see. Up all the way. Hopefully that'll be loud enough. We're about to get started. I don't want to wait too long because in case you guys didn't realize, like if anybody looked at the video, we're not going to get anywhere near through the chapters for this. So I mean, we'll get to 10 and we'll get to 6 and 8 for sure. That's part of it. So I still have time. We don't have an hour to do. All right, so I'm going to go ahead and get started on this so that we can get moving. We're going to start with genetics.
start with chapter 6, because that was the thing that I feel like everybody had the most problem with. Then we'll go to 10, which is the second problem. And then we'll go to, I'm going to go to 8 in between because it just comes after 6. It makes sense to me as part of it, basically. But anyways, that's how we're going to do that. And then 9 and 25. And hopefully, if we can, this 13 and 14, but we'll see. All right. These are kind of set up in, I want to say, bite-sized concept sections or whatever. So what we're going to do is, I hope you guys can hear me online. I don't have any indication whatsoever. If you guys can hear me, sorry. Yeah. So we're going to talk about DNA first, and then we're going to talk about like replication, transcription, translation, operons, and mutations. That's how this is going to go. All right. So with DNA, we should know for bacteria, their chromosomes, they have one chromosome that is circular, and that's it. Whereas we know when we look at our own chromosomes with those little X's lined up, that is linear, and we have many of them. So that's eukaryotes. The difference between phenotype and, great, glad to hear it. I was so worried about it. That other computer. Phenotype and genotype. Your genotype is like your genes, right? So that's like you got one gene from your mom and one gene from your dad. Everybody has that, whether you're good with the relationship or not, doesn't matter, right? So you have a gene from a mom and a gene from a dad. So if they are different, typically one is going to be dominant over the other and get expressed. And we won't even know we are carrying around the other one. The one that's being expressed gives us our phenotype. That's what we see in the individual. Whereas the genes are what we're actually in our genetic information. You can still pass that information on, whether you're expressing it or not. But what you express is your phenotype. Okay. So RNA and DNA and what their differences are. DNA is the one that we're probably most familiar with. Most people are aware of what DNA is, but that's our genetic material, right? Whereas with RNA, its function, mRNA, working copy, that's the working copy that we make of a gene from DNA to make a specific like protein, right? I don't want to be rude or anything, but you guys know that these are online, right? These slides are available. Yeah, yeah. You don't have to take pictures of it. So yeah, this is all the same as what's online. So yeah. Anyways, I just don't want you guys to like feel like, you know, you're missing something if you don't take a picture of a slide or anything. If you want to take pictures, don't let me stop you. If you want to take a video, don't let me stop you. This is one of those times that I want you guys to get what you need out of it. But yeah, I just want to remind you that that's on there. Right, so working copy, mRNA, that's like the gene that we're going to actually be reading that part. We go to the book, the compendium of everything that has all of the genes at all. Then you go in there and make a copy of just the one thing you're trying to copy out. Like a recipe book, we're copying out the one recipe. That's mRNA. tRNA transfers the amino acids when we're actually reading through the mRNA through translation. And we have ribosomal RNA. That's what actually makes up the structure of the ribosome. So those are the three kinds of RNA and what like kind of their functions are. Whereas DNA, it's just the genome. That's all it really is. Has the genes. Another way that they differ is their 5-carbon sugar in their backbone. Both of these guys, you know, are sequences of nucleotides, right? And nucleotide, which is down a little bit further, is going to be a nitrogenous base. That determines whether or not it's A, T, C, or G. Do not worry about a purine versus a pyrimidine, okay? That's not on the text. Leave it. That's not going to be there. Just know that is a nitrogenous base. And what does nitrogenous mean? It has a lot of nitrogen in it, right? That's all that you have to worry about there. They are 5-carbon, nitrogenous base and a 5-carbon sugar, and then a phosphate group. So the sugar, 5-carbon sugar, and the phosphate are going to be repeating over and over again as the backbone, sort of like, sorry, I don't have the pen thing to draw, so it's harder with the mouse, so bear with me. So if this was your piece of RNA or DNA, all of this would be sugar, phosphate, you know, over and over again, repeating. And then sticking out from that structure from the sugar actually will be our nitrogenous base. So this guy is going to determine A, T, C, or G. The backbone, same for all of them. Okay. So with DNA, that backbone, that sugar is deoxyribose is what it's called. The 5-carbon sugar. With RNA, we have just ribose. And what's the difference? We just removed an OH group. That's the only difference there. Okay. That's why deoxy. We deoxied it. Okay. All right. And then we have with DNA, we typically work with it as a double-stranded entity, however you want to look at it. It's double-stranded, whereas RNA, we're typically working with that single-stranded. That's because we're going to be reading it pretty much right away. It doesn't need to have like a matchup to help keep everything stable and to make copies and replication from. Really, that's why DNA exists as 2 strands, is to help with that replication process when we need to make new cells and stuff. But anyways, DNA double-stranded, RNA single-stranded. The other thing, DNA uses thymine. So when we say A, T, C, G, T is for DNA. RNA, it's the same exact things, except instead of T, we have U. That's uracil. They're slightly different structure, but they're very similar in nature. It just helps with the actual function of the mRNA versus the function of the DNA. You don't really need to know anything about it. Just know that it is different and that is equivalent to the T. Okay, so A, U, C, G for RNA. Otherwise, they are pretty much the same thing. So that's that for RNA, DNA, and nucleotides. We do refer to DNA as being like the double-stranded DNA, as being anti-parallel. So we've got 2 strands, and it means that those 2 strands, we know how that works, right? It's like a ladder, and we say one strand goes in one direction, and the other strand goes in the other direction. What does that mean? So we've talked about this up here with our sugar and phosphate groups up here, right? Over and over and over again. Well, the orientation, because you know, some sugars have like 2 OH groups on like one side and none on the other side, depending on their molecular makeup. Well, it's the same thing for ribose or deoxyribose. They have not quite, mirror image of one another. So that makes a difference as far as where there are available spots to bind to and interact with, where those OHs are or where they aren't and stuff. So it's like if I were to take like, I don't even know, a piece of paper and like write on one side of it, like A, and on the other side also A, that if you flip it over, it might look the same, but you had to flip it over to make it look the same. These guys, essentially, looking at them at that molecular level, aren't flipping. So they're still kind of wrong side over, if you will. So they're running in opposite directions because the sugars aren't like perfect symmetry. So, but the chemical direction of the sugar phosphate repeating and repeating and repeating maintains itself. But yeah, it's all dependent on the sugar, basically, is what I'm trying to say, because it's not totally symmetrical. So we would go, and we say 5' to 3', y'all sick of hearing me say it probably. Our direction of movement is going to be 5' to 3'. That is literally just one of the carbons on the sugar. It doesn't matter how that we determine that. You guys don't need to worry about that. But they're just number that, chemists like to do that. Say that this carbon we'll say is number 1 because it's blah, blah, blah bond here. Well, it's number 5 carbon, which is the last, we would say the last carbon. It's free on the 5' end. And the 3rd carbon that they decided was number 3 is free on the 3' end. So that just means what's going on in how those sugars are oriented, because they are not completely like mirror image within themselves. So yeah, so the other matching strand, A to T and C to G, with that ladder that we're making, that match up one another, that one is going to be 5' over here and 3' over here. Again, it's just due to the orientation of the backbone, and that's sugar primarily. That's all. Sugar orientation determines 5' to 3'. And it's anti-parallel because they run in opposite directions. That's the 2 things that you really need to know about. I'm not going to make you write a friggin' essay about 5' to 3'. I will want you to be able to recognize in multiple choice what anti-parallel means, right? The backbones are running in opposite directions. Okay. That's what you guys need to know. Ooh, okay, DNA replication. This is going to be always in that 5' to 3' direction. Because of how those nucleotides are oriented, we have to add them on in a certain orientation. So it's always going to work 5' to 3', and we always have to have like a primer there to work off of, because the polymerase can't add to nothing. So it'll add on to some sort of primer. It's usually RNA, but we'll remove it later and replace it. So it's not too important, that part, but we're always going to move into 5' to 3'. So whenever we start to make copies of our DNA and we're opening up this, and I'm going to have a video that'll explain this better than my words. So I just want to kind of word it out first. You have your double strands. And when we have your double strands, because I can't draw very well with this freaking mouse, as it opens up, Jesus, right? If we are making our copy, and this is 5', and then we're going, that's a 5, not an S, and we're going in towards that way, we can just keep going. That's going to be 5' to 3'. Yeah, let's just keep while we're making our new strand. But the other strand can't do that, right? Because it's in the opposite direction. So it can't go in towards the fork. It has to come out from the fork, the opposite direction. Yeah, and that makes sense, except the out from the fork is constantly reopening. So how do we deal with that? So as it opens up, we will make segments and read the segment, and then it'll open up a little more, and then we'll go to the next opening and we'll make a segment and eventually join all of those together with DNA ligase. Remember, ligation, to ligate something is to join it, seal it, basically. So DNA ligase will seal those fragments up. Those fragments are called Okazaki fragments. The strand that can go straight on like that, that's nice, that's easy, that's quick, that's the leading strand. And the other one is the lagging one, because we have to be made in parts. You have all this crap you have to do in order to make it. So that's the lagging strand. It takes a little bit more work to deal with it. So I will see if this will play on this computer. It worked on the other one. So let's see if it works on this one. This is as loud as it goes, unfortunately. I don't have actual speakers. I might have closed caption though. Okay. Oh. Okay, I think it's done for real this time. I can't see, like, when those videos play, I can't see like how much time is remaining. So I have no idea like how much longer I'm gonna have to sit through it. So if you guys are online, I don't know if you could hear the video or not, but I will tell you guys, if you open up the actual PowerPoint, and you guys are welcome to do this, of course, too, that you can access, you should be able to access these videos that way. And if you can't, it is from the textbook. So that should help, I feel like. So there, it is available. Okay, this next video is just another kind of, it's the same thing, basically, but I'm gonna play it anyways in case somebody else resonates more with this than the other one. I would say I was gonna play it.
Replication some hydrogen
Okay, I think that's that. Yeah, so I do want to point out, it's talking about like it can only add on to the 3' end, right? That makes sense, because if you're moving in a 5' to 3' direction, what's at the end of that? The 3', right? So I don't want you guys to think that that is something different. It's not. If you need to draw out a line and say this is 5', this is 3', do we add on to the 3'? Well, yeah, because you have to move from 5' to 3'. Okay, that's the direction. The orientation is a different thing. Okay, so I hope that helped you guys. Yes. No, absolutely not. Yeah, just know that it is DNA polymerase and you're good. And that is an enzyme, right? ACE. Anytime you guys see that enzyme. So does that help people understand kind of the replication stuff a little bit better? Words, it makes it so difficult, I feel like, explain it with words and me trying to draw my crappy drawings. So I hope that the videos are a little bit more helpful to you guys. All right, we'll move on to transcription next. Now, we have DNA replication, right? That's just making copies of DNA. Why do we need to do that? Anytime we make a new cell, it needs to have DNA in it, so we have to make a copy. That's why. RNA transcription, this is when we have that big old recipe book, and we're going to open it up, find the recipe that we want, and only copy down that recipe. So here's our recipe that we've written down. That's mRNA. That's our working copy of that recipe. The actual gene stays in the nucleus with the DNA. Doesn't go anywhere. We don't take that out. Only the copy of it, the mRNA. So we already talked about the 3 different kinds of RNA. So I'm not going to touch on that again, but we'll get to their uses in translation. Okay, so transcription, which you can already see. Recipe book, making a copy of the recipe you're interested in. Well, how would you do that? By writing it. A scribe is somebody that writes, right? Transcription. We wrote that down. So now we wrote down that recipe. This is going to have to happen in our cells and eukaryote cells in the nucleus, because that's where we keep our genes. So we have to go in there to do this because the DNA does not come out. So, but in bacteria, they don't have a nucleus at all, right? So that we say that's in the cytoplasm. It's really in the nucleoid region, which is just where the DNA is hanging out. It's just the area where that's hanging out, okay? They don't have a true nucleus though, so that can happen just out there. This is just reminding you guys, A to T, G to C in DNA, but then whenever we're making RNA, switch that T with the U. Still binds with the A. Okay, so that's transcription. I have a little video for that.
During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the prolific region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand. As the complementary bases pair up, the RNA polymerase links nucleotides to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete, and the RNA polymerase, the DNA strand, and the messenger RNA transcript dissociate from each other.
Okay, so that one, I feel like that one's pretty straightforward, right? For making mRNA, you want your working copy. You only need it to be single-stranded. That's nice. That helps it go a little bit faster. We match up those A's and U's and stuff like that. We're going to match that up as we go, and then it just is done and it falls off. 3 steps: initiation, elongation, termination. Same steps for translation, which we're going to talk about next. So remember, we had our DNA that we replicated because you fell and scraped your knee and you need new cells. Made replicated DNA. Now that cell is ready to make parts and stuff like that. So it's going to go into the genes and make a copy from its recipe book, right? That's the whole genome. And make a copy out of the ones that we want to do. So we'll take that. That's our mRNA. And that process was transcription. Now we take what we wrote down outside into the cytoplasm, to usually along the endoplasmic reticulum in our cells, right? So this is ribosomes now. They usually hang out on the endoplasmic reticulum. So anyways, we'll take it over to that and we'll start cranking out some protein. We're going to start turning in what we wrote down into the product. So it's sort of like translating it. It's not going to be a written down thing anymore. Now it's going to be the product. So we're translating that message into the actual product that we want. Okay. If you look at DNA, you can separate it into 3 nucleotide segments, like AAA. All right. Those are called triplets in DNA. However, in mRNA, our messenger, our working version of the gene, the 3 nucleotides are going to be called codons. And those codons, each one codes for a specific amino acid. Now we have 20 amino acids total. And so some of them code for the same amino acid. That helps for if you have like any mutations in there to try to help maintain what amino acid you're coding for. That's why it's called a wobble effect. But anyways, that's what the codon is for. It's going to code for the amino acid. But how do we get that going? You say it codes for it, but what is that? So what we do is that codon, that 3 nucleotides, will match up A to U, G to C, whatever, those 3, with that tRNA anticodon. So it's like the opposite code on the tRNA. If they match up and they're complementary to one another, then the amino acid associated with the tRNA will be added on to that growing polypeptide and eventually protein. Okay, there's a video, don't worry, that explains it better. I like the videos, but that's the idea behind that. So the ribosomes are helping all of that happen and helping everything move over as we are going from amino acid to amino acid to amino acid from those 3 nucleotide codons. Yes, okay, so I'm happy with that. Let's watch the video. This one is one of the other ones that I can't see the time on. You don't need to know the site names, okay? You don't need to know the site names, but I feel like the visualization of this helps a lot with understanding what's going on. So this part here, like, helps really visualize what's happening. Codon to anti-codon binding, and then it shifts over. And while it's doing that, those amino acids are going to get linked together. That bond is called a peptide bond. All right. Like I said, I feel like less complicated when you see the video. All right. The next thing is the operons. Now, operons, they're only in bacteria. That's the number one important thing to know about that, right? Operons only exist in bacteria. And what they are is just a regulated set of genes that all work together to do a similar thing. So genes that are all going to be involved in breaking down lactose, which is what we see in the lac operon, or genes that are all going to be involved in making arginine, which is an amino acid, which is another type of operon. Stuff like that. So that is the idea with an operon. All those genes would need to be made together. In bacteria, they can just all be made in kind of one controlled, in one sitting, and made in one transcript, and then make the protein off of that. But yeah, ours doesn't work that way. That's not that important. So there's two kinds of operons, the repressible ones and the inducible ones. Both of them are controlled by a repressor. Now that makes sense. Think of the repressor as like the cop that like stands out in the road and lets you go or not go for the church traffic, okay? So based on whether he's out there or not determines whether you're driving through or not, okay? So he's the repressor. And we can control whether the repressor is out there or not based on each type of operon. So we'll get to them individually. Repressible. If it is repressible, yeah, you hear that noise? I don't know. I don't know what that is. Sounds like a baby or kids or something playing outside. Anyways, repressible. If you can repress something, that means you can stop it, right? Like silence it, basically. To repress it, it has to be on. So typically, repressible operons are always going to be on, and you can repress them. They're repressible. They're able to be repressed. So they're always going to be on, but you can turn them off, and usually that's going to be due to a buildup of your product. And what I mean by that is, if we have an operon where it's always on, and it's always making pants, okay? We're making pants at our factory, our operon factory in the cell. And we're spitting them out and spitting them out. People are just buying them off the shelf, so we're keeping up with the demand and making pants, right? Well, let's say summer comes around, and now people want to wear shorts. They're not buying as many pants. And so whenever that happens, we start having a back build of pants at first, right? So the factory goes, hey, we don't need to make quite as many pants now. We should probably do something about that. That is as though if we had the buildup of the pants happening there, when they realized that the pants were there, if the employees brought the pants to the manager would be the repressor here. The manager would say, okay, time to stop production. And he would halt production And so we needed more pants again. So he's going to change his mind based on whether or not there's too many pants there, right? As long as they're getting used up, we're not affected by it. They're going away. Nobody has any to go show the manager. But once they start back building, we get extra built up. Now we've got too many. And then we start bringing them to the manager saying, hey, too many. That way our repressor, which is a protein, when it sees the pants, which is the product that we're making, yeah, that product and the repressor interact with one another, it changes the repressor, so it's going to go stop the gene. Because we don't need it anymore. We have too much. Until we need it again. All right, that's repressible. We're always on, and now we're stopping it. Inducible, the best example we have for this one is the lac operon. The lac operon, go figure, involves breaking down lactose. A lot of bacteria are going to rely mostly on glucose. You guys were studying about your metabolism stuff, and probably that's no surprise. Yeah, glucose, the ideal kind of sugar to get your energy from. So they have these operons in place that if they see lactose, then they turn on. So they're off. Those genes are off. We don't need them all the time. We only need them when lactose is present. So they're always off. And then when lactose comes around, then they will turn on those genes to start breaking down lactose. So a good example of that is like people calling, maybe stores calling up the factory and saying, we need more pants again. You know, that was a different scenario, but that's the idea with that. So the repressor here is just going to be sitting on those genes, waiting like the cop in the middle of the street, just sitting there waiting for, you know, someone to tell him to get out of the way so the traffic can go through. And so what happens in this case is the cop, the repressor, is going to be activated to move and clear the way for RNA polymerase when lactose is present, and only when lactose is present. They bind together, they interact. It's like giving him like an ice cream cone, and he's like, I'm gonna go eat this ice cream cone. And now he moves out of the way and everybody can start driving again. Okay. All right. That's the best I got on operons. Alrighty. The video sucks. We're not gonna watch it. It really sucks. Like, it's way too much information. All right, mutations and gene transfer. This is two different things, but they fit on one slide. DNA mutations. We can even have point mutations. That just means like one A got changed into a G or something. So one nucleotide change is a point mutation. Anytime that we have any kind of mutation that leads to a different amino acid than what was originally there, that's missense. So kind of think of it as like a misfire. The right amino acid didn't get there, but there is amino acid, okay? Whereas we're comparing that to nonsense. Nonsense meaning we ain't getting an amino acid. It's just no. So that's when we have a change in the sequence that causes a stop instead of where there should be an amino acid. Missense, different amino acid. Nonsense, stop. Okay? Shouldn't be there. Silent mutation. We changed a nucleotide, but it didn't change the amino acid. Like I said, some of them code for the same amino acid. So that would be a silent mutation. We don't care. We wouldn't see any difference in the protein. So your body doesn't really care about that. A back mutation is if you had a mutation on a gene that was mutated previously. So we're going back to wild type. Okay. So if the original was green and we had a mutation that turned it blue, but now that mutated and turned it back to green, that's a back mutation. Okay. And then we have frame shift mutations. This is a little bit different here in the example that I'm showing here, AAA, GGG, CCC. Take out that third A. That's what that line is going to represent, okay? Just take it out completely. But we still have to read in threes, right? The body doesn't know differently. So this would be like taking out that A and then shifting everything over one nucleotide after that. You could see, even like looking at this, obviously, very different sequence now. Going to code for very different amino acids as well. Another way you can think of it is if you take like a book, like a novel, you're a good couple chapters in. If I take out one letter but maintain the shapes of the words. You know, this one's 5 letters, this one's 7, whatever. And then shift everything over by that one letter. Would you be able to really read that anymore? No, it wouldn't make any sense. That's the idea here. Frame shift mutations are very, very serious, as are nonsense mutations. Okay. All right, so those are the mutations. Bacteria have the ability to transfer genes between the cells. We talked about this whenever we did our transformation lab. The 3 different ways that you can transfer between cells, which is called horizontal gene transfer. We have transformation, obviously, we did that in the lab. That's going to be picking up DNA from outside of the cell and putting it into the cell. Transduction, that's going to be viruses, basically. So bacteria infected with a phage, that phage inserts its DNA into the bacterial chromosome. That's called a prophage then, by the way. That prophage is in there, but it has extra genes in it that help the bacteria make toxins and become more pathogenic in general. So that's the idea with transduction, and that's what we called lysogenic conversion, if you guys remember about that. That's the same idea. Conjugation, like a conjugal visit, guys. Yeah, so what conjugation involves sex? Yes, it does here too as well. Sharing our DNA via the cell-to-cell sex pilus. So it's just a tunnel between the 2 cells that forms, and they can shoot DNA between them. All righty. This kind of sounds, does actually, now that I think about it, sound a lot like what people do too. Yes, yes, yes. Okay. Now we're going to just move into chapter 8, since it just, I feel like, segues into that. Gel electrophoresis. This is a method that we use to look at the size of DNA pieces. Even if you're not that interested in the actual size of the DNA fragments, this will at least give you a sense of how that DNA fragment was affected by whatever you cut it with, for example. You can compare that from person to person or creature to creature. DNA itself has a negative charge to it, right? Backbone has the sugars, that's a sugar, whatever, and phosphates. We already know phosphates are negative. We keep saying it with the ATP. So sugars and phosphates, it's got a negative charge to it. And so it's going to move to the positive end of the machine or whatever. Okay, cool. So it's going to move to the positive. In general, we measure the size of DNA fragments in base pairs, right? How many base pairs long is that piece? BP, base pair, or KBP, kilobase pairs. Kilobase pairs is a number times 1,000. So 5 kilobase pairs is 5,000 base pairs. Okay. Smaller bits of DNA are going to move faster through that gel gauntlet, and the larger ones will get caught up. Think of like if you're playing football and there's a whole line of linebackers there. The little guy's going to be able to squeeze through a little bit more easily than the other big guy. He's going to get caught up. So there's that, you know, sort of idea going on there. So the smaller ones will be at the end of the gel. The larger ones will be where you started, near the top of it. Okay, and everything in between will be in between. The next one, PCR, polymerase chain reaction. Each cycle doubles the number of molecules that the previous one had ended with. So what does that mean? Well, if we start with one piece of DNA, one double-strand DNA, after one cycle of PCR, we're going to have 2. If we had 2 double-stranded DNA, after another cycle, we're going to have 4, right? Because each one will make 2. There will be a question, I don't remember if it's on the lecture test, but I know for sure there's a question about this, about how many you'll have after a certain number of cycles. So be aware of that. That's definitely going to be on the lab exam. So if you need to draw out, like I had said to my other class when I was giving this, if you need to like draw out the line, like a picture of how it's working, that's how I do it. I'm not good with math. So I just like say, okay, this is cycle 4, we'd have this many based on the lines that you draw. Cool, cool, do what you need. Right, so steps that are involved in PCR, pretty simple here. Denature, which just means breaking apart the DNA strands with high heat. The heat causes those hydrogen bonds between the 2 strands, which that's all that holds them together, to weaken and break apart. They're pretty weak bonds. Next, we have annealing. This is just those primers. We already saw in that other video that DNA polymerase can't build off of nothing. Additionally, the primers match up pretty well to specific sequences. So now you can target very specific genes to see whether or not that gene is present. So that's how we, diagnose things, actually. So denature it, break it apart into separate strands, anneal primers on there, and then build off those primers, elongate. That's all that we have going on. And we'll do that over and over and over and over again for like 30 times. You can get billions of copies going through that. All right. Cloning, really here what I'm just talking about with cloning is using like the DNA of like one species into another species for some sort of gain. It's usually for our benefit, right? We're not just doing, sometimes we're doing it just for science, but hopefully for a gain of some kind. Knowledge is a gain, I guess. Anything that you're going to put your gene into to get it into the animal you're getting it into, like a person or a bacteria, whatever you're using, here are the vectors you can put your gene into. Plasmids. We already learned about a plasmid. In the transformation experiment, we saw that GFP gene. That was the gene that we had inserted into that plasmid. That was the goal gene. You could just as easily have put a gene for human insulin in there and made that bacteria make that instead of GFP. And that is how we actually get human insulin. I mean, yeah, for your shots and stuff like that. That's where it comes from. Anyway, so that plasmid, that's pretty useful. And we can control a lot of what's going on, like including that antibiotic resistance that we saw with our different plates. We have viruses. We can engineer viruses to carry genes instead of their deadly genes. That's how a lot of gene therapy works. And then we have artificial chromosomes, which is what it sounds like, designing a chromosome from scratch, essentially. All right, transgenic means we have changed the genes. GMO, genetically modified organism, same thing, okay? Anytime that you change the genes of the thing, GMO or transgenic, same. The other last thing, which actually was talked about in the beginning of the chapter, but on your study guide, it was at the end of this part, restriction endonucleases. These are just enzymes, ACE, right, at the end. These are enzymes usually made by bacteria to get rid of like foreign DNA that are in their environment. But we have taken them and used them in science because they cut at very specific known places that are unique. They're palindromic in nature. You don't see that a lot naturally, like all over the place. It's rare. So they will only cut at specific palindromic sequences. There might be one that recognizes one palindrome, another that recognizes another, and so on and so forth. They cut in a way that leaves kind of a single-stranded end on each side. And we can do the same thing with our target gene, like our insulin gene, put the same thing through genetic engineering on the ends. So we can now stick it into the plasmid. It'll match up at those sticky ends, okay? So that is a very useful thing. You'll cut at specific places, create sticky ends. Specific place for your binding is a palindromic sequence. Awesome. Okay. All right, chapter 9. This is it, getting into nutrition now. Doing good on the time. So I'm gonna try to just truck on through this one so we can get to 10. Okay. Because I feel like, do you guys want 9? If you had to choose 9 or 25? 25, you think? Okay. Chapter 9 is primarily vocab words. Okay. So hopefully we can get back to it, but if not, let me get out of this. Sorry, it's faster if I do it this way. Okay, 25, let's just swing through this one really quick. Now, 25, if you guys are looking at the questions on the study guide here, let me just tell you right now. If you're looking at it and be like, I don't know, we didn't really talk about this. We didn't really talk about, I don't remember this, whatever, blah, blah, blah. Number 1, nope. Number 2, nope. Number 3, kind of. We'll talk about a little bit. Nope. Number 4, nope. Kind of number 5, right? Because that's talking about the carbon cycle and the nitrogen cycle. That's all that's talking about there. 5. Number 6, duh. If you need me to tell you that it's in the oceans, you got some problems. Okay. So it's in the oceans. What is Potable water, not going to test you on it, but it just happens to be pathogen free. That's what that means. It doesn't have to be clear, just pathogen free. Kind of talked about cleaning water, so that number 8 is good. Kind of about number 9. You guys know what microbes do in food. You don't need me to tell you about that. Number 10, ways to limit microbial survival or growth. That's just like refrigerating or heating something up to get rid of organisms. Don't read too much into these questions, guys. And then number 11, forget it. Okay, does that help a little bit? It should, because I just eliminated like 6 concepts out of there. All right, this is what I kind of need you guys to know about chapter 25. Chapter 25, the carbon cycle. Carbon dioxide in the atmosphere. The plants and other photosynthetic organisms, including photosynthetic bacteria, are going to use the carbon dioxide from the atmosphere and fix it into organic molecules, right? That's what they're doing for themselves. Then other organisms, the heterotrophs, which these terms are from chapter 9, they're going to go through and break down those organic molecules for energy and exhale as a waste product, which we'll talk about in number 10 or chapter 10. Carbon dioxide is waste from that, right? That goes back in the atmosphere, and then what takes it back up and goes back to the cycle? Photosynthesis. That's the carbon cycle. Don't read too much into it. Carbon dioxide, photosynthesis puts it into organic molecules. The other organisms that aren't photosynthetic are going to use those organic molecules for themselves and have carbon dioxide as a waste back to the cycle. Okay. Nitrogen. It's important between bacteria and plants. Plants cannot get their nitrogen without the bacteria. Bacteria break down all sorts of nitrogen sources into all sorts of nitrogen products, and as a result, allows the plants to use those products. The processes that they go through: nitrification, denitrification, ammonification. Guess what? For this, I do not need you to know what those are. Know that they are important to nitrogen cycle. I don't need you to tell me which one is nitrate to nitrite, and then nitrite to, ammonia. I don't need you to know that, okay? Not here. We talk about it again in the lab later at the very end, but don't worry about it right now, okay? That's that. That's that for that, okay? Water. Potable means pathogen free. That's what the question was asking. Freshwater treatment and wastewater treatment, we're going to have sedimenting. We're going to have filtration after filtration. The difference with wastewater, we're going to have sludge there. So we have to break that down, usually by bacteria that can degrade it. Then treatment with chemicals. Aeration kills all the anaerobes, right? Because they can't deal with the toxic oxygens. So aeration, then chemical treatments and other filtrations, and then storage of the water or putting it back to the environment. How do we make sure that it's safe for the environment? What we do is take samples and filter them, put the actual filter onto an agar plate, just like we have, and then count how many colonies show up. We can even determine which of those colonies are coliforms. And coliforms are like fecal bacteria that aren't necessarily harmful, but if they could survive the treatment process, the pathogens could too. So you need to look at how you're treating your stuff. That's that. All right. The food. Do you need me to say these things? I don't think so. Okay. That's chapter 25. Y'all worried about too much. All right. I'm just saying, this should be very comforting to you guys. I mean, I'm serious. I'm not going to test you over more than what I'm talking about. Now, am I going to ask you the exact same thing? Absolutely not. But if you understand these concepts, writing it down and having it memorized is useless. Don't do it, guys. I hate people that memorize things and think that's enough and come to me and complain that they didn't do well on the test. If you don't understand it, what's even the point of doing it? So so my test expects you to understand these things that I'm saying to you. Then you will be able to do well on the test. Don't just copy and paste. All right, this isn't, freshman class. So, right, so moving on. Metabolism. This is the big one, yeah? We got time to look at that. Metabolism is all chemical reactions in the body. There's 2 basic kinds of chemical reactions: when you're breaking things apart and whenever you are building things back up, yeah? We do building up if we have damage or if we want to make new cells, whatever. We do breaking down if we want to get energy out of it for something. Usually that's what's going to be going on, or need the parts of it for something else. All right, so anabolism is when we're creating bonds. Catabolism is breaking bonds. And I don't have any good way to memorize anabolism, but catabolism to me is obvious because cats are pretty destructive. I'm not saying dogs aren't, but cats are definitely destructive. So that's how I remember this cat is able to destroy catabolism. Okay, breaking things apart. All right, enzymes. These are proteins that help chemical reactions go, right? We say overcoming activation energy. That's literally what's happening, but they're just helping the reaction go. They're proteins. All right, they can, a lot of them have to work with parts that work together, not just by themselves. A holoenzyme is the term for the whole enzyme. Okay, the whole one that works and functions. It consists of the apoenzyme, which is just the protein portion, plus a cofactor. Now, a cofactor can be inorganic, like a metallic ion, which we would just call that a cofactor, or it can be an organic molecule. If it is an organic molecule, like a lot of vitamins are, if we have to interact with those things in order to function, then we call that a coenzyme. Turns out that a lot of the electron carriers that we're going to talk about here in a second, those are really coenzymes. They are organic molecules that are going to interact with enzymes. Really all they are. Okay, those are the terms for that. The 6 types of enzymes. The only one that I feel like is particularly difficult, I mean, of course, the other ones are flashcards. Yeah, this is a flashcard, memorize and slap it down kind of a thing. Oxidoreductase is the one that I would understand, right? That's an obvious one. If you didn't see that coming, I don't know. Oxidoreductase, what are we talking about there? Redox reactions, yeah? You guys remember them from chemistry? A redox reaction, reduced or oxidized. You always have one doing one thing and the other doing the other thing. Always have one and the other paired. So an oxidoreductase is an enzyme that's involved in that. Another way you can think of it, anytime that we are moving around electrons or have electron transfer, that's oxidoreductase that's going to get involved. Okay. All right, here's how I remember it with my this oxidation reduction. Leo, I'm so sorry, this is going to take me a minute to type it or write it out. Leo says GER. Now you also can do oil rig. Oil rig is fine, but it's the same exact thing. So Leo, lose electrons oxidized. Okay, even if you just remember Leo, you got the other answer there. It has to be in a pair, right? So lose electrons, you're oxidized. If you gain electrons, you're reduced. That's it. Oil rig. Oxidized is loss of electrons, and you get rig. So same thing. So that's how I remember which one is oxidized and reduced. Sometimes, I'm not joking, like I say sometimes, I mean almost every single time that I'm talking about a reaction that involves electron exchange and somebody's asking me, was this molecule reduced or oxidized? I have to write this out. I don't know it like just by my heart or whatever. I've been relying on it too much is probably the problem. I rely on it too much, but that's how even I figure out which one is reduced or oxidized. I'll write that out. I'll be like, okay, did this lose the electron? Okay, yeah, this lost the electron, so it was oxidized. That sort of thing. All right, these other ones. These are some terms that I would do flashcards for just to be aware of what they are, just as like a term thing. Transferases move groups around. Isomerases change shapes. Liases, double bonds are involved. Ligases, this is, oh, we've already talked about DNA ligase, right? What does it do? It's going to join bonds, yeah? And when it does that, it's going to do so by giving off water as a product. Hydrolases break bonds and use water, break water apart in order to do that. So you can see ligases and hydrolases kind of similar, like opposites from one another. They work together. All right. Next, we have the regulation of these enzymes. We have, we've already talked about the operons and like that sort of stuff, and that we can have products and all sorts of things binding with proteins and that can go interact with the DNA. That happens in our cells too. It's just not operon. We don't call it that. But we have similar ways of regulating our own genes at the genetic level. That, whatever, cool. So, but the ones that are happening at the actual protein level and controlling it there, that's going to be competitive or non-competitive. When an enzyme has something that is changing from A to B, the thing that is working on A, that's called the substrate. It's changing the substrate, right? Or helping the substrate do its thing. So if a competitive inhibitor, the thing that's going to inhibit, if that binds at the same site that the substrate was at, that's called competitive. It's competing with the substrate at the active site where the reaction was happening. If it's competing for the active site where the action is happening, then that is competitive. Non-competitive inhibition, maybe down that whole line, you have product buildup, like the pants from earlier. The pants might come over and tap the enzyme on the back, and when it's tapping or sticking to the back of that enzyme is like turning around. Is it changing its shape? That's essentially it changing its shape, okay? See, if the pants changes its shape, no longer can it interact with the substrate. So, but it's not binding at the active site, right? At a different place, and we know how proteins like to change their shape whenever they interact with other molecules, right? That's the difference between competitive and non-competitive. Does that make sense to everybody? Cool. That's pretty easy one, I feel like. All right, respiration. Here we have aerobic, anaerobic, and fermentation. Those are the three ways that organisms can get their energy. Energy in the form of ATP, right? ATP has energy because those phosphates that are highly, highly negative are going to push against one another. And that creates a lot of tension, and tension equals energy. So great. So we've got that. We understand about ATP and why it's important. As far as what ATP is, just real quick, since I'm talking about it, adenosine triphosphate is what it stands for. We have an adenine, which is the same nitrogenous base. That's the A in DNA and RNA, by the way. Ribose, yes, the same sugar as what's in RNA. And then 3 phosphates stuff on there. So it's basically a nucleotide with a couple of extra phosphates. All righty, so that's what ATP is, and the energy is in those phosphates. Aerobic respiration is going to require oxygen, right? Air. That's going to make the most energy out of all possible things. Anaerobics, they use something other than oxygen at the end. It's going to be nitrate or sulfate, typically. And then fermentation, we just go to glycolysis and then turn right back around and just keep doing that over and over again. All right, in order for all of this to work, we have to have these, they're coenzymes, but they're mostly, what I want you to know them as is electron carriers. That's NAD, you don't need to know what they stand for, NAD and FAD. When I have them as NAD and FAD with the little pluses, they don't have their electrons on there. During this whole process, they're going to get electrons added on in the form of hydrogen atoms with the electrons. And when that happens, they're gaining an electron, so that's reduced. So this is where it comes back around. All righty. That's the setup. This is all you need to know. Do not go beyond this. Okay. Glycolysis is the first step. We're taking glucose. It is a 6-carbon sugar. We already know it. We're going to break it basically in half. The product, the 3-carbon pyruvate, we're going to have 2 of those at the end. So 6 carbon, 2 3 carbons. Taking glucose and turning it into pyruvate is the goal of glycolysis. That's the main goal of glycolysis, right? And in fact, I've written the main goal of each of these steps in red on the slide. Main goal, pyruvate. We also make a little bit of ATP. Turns out about 2 net. You don't need to know the number. And we also have a little bit of NADH that we will make at this point, just during the process. They happen to be nice little products that we get out. They're not the goal, but it's nice little, icing on the cake, if you will. Yeah, that's glycolysis. That's what you need to know. In the middle between glycolysis and Krebs, this isn't as important, but it is where some carbon dioxide and an NADH come from that helps with the whole process. We're going to lose a carbon off of pyruvate and turn it into something called acetyl-CoA. Not an important step really, but that is what happens before we go into the Krebs cycle. Okay, lose that carbon, it comes off as carbon dioxide. The electron that was associated with that bond, we made NADH. Okay, that's really all that happened there. Now our little 2-carbon acetyl-CoA, we're in the Krebs now. We're going to merge that thing, that 2-carbon thing, in with all these random other organic molecules. And we've seen it, right? The big circle with all these organic molecules like citrate and oxalate and all of that crap. 2-carbon acetyl-CoA, we're going to start interacting with those organic molecules. And then start taking carbons off, start stealing electrons and interacting with all these different bonds to change it up so we can steal more carbons and whatever to get electrons. The goal here is to steal as many electrons as possible. We hold onto them in our electron carriers, NADH and FADH2. The goal of the Krebs cycle is NADH and FADH2, electron carriers. Okay, that's the goal. In the process, breaking down and changing up all these things, moving them around, we'll make CO2. This is where most of the CO2 is made, but you don't need to know that. We do make CO2 though. So if I ask you in which steps maybe CO2 was made, that's the type of thing that I'm talking about. There might be questions that come from this that are answered in here, but aren't direct questions in here, right? So that's the type of thing. Like which state, of the choices has CO2 made? All right, cool. But we make CO2, we make, again, a little bit of ATP. With each pyruvate, we get 1 from the Krebs cycle, so 2 overall from 1 glucose, yeah? So 2 ATP from glycolysis, just on the way. 2 total ATP from the Krebs from both of our pyruvates that went in. Okay, you don't need to know the numbers. I'm just kind of setting it up for comparison. We've got all these electron carriers. We broke down glucose, made pyruvate, split it apart, shoved it into the Krebs, got our electron carriers. We're now at our goal so far. What are we doing with all of this? We're putting our electrons into the electron transport chain or electron transport system, same thing, to make ATP. In this step, we are going to make, just in this step alone, with 1 glucose molecule, 32 to 34 ATP molecules. You see the difference there? When somebody asks you which one makes the most ATP, you shouldn't be thinking like, I don't really know which one I have. This should be immediately electron transport chain. This is the big daddy, it's the goal, okay? I'll tell you why it's so important here in a moment. So we've put our electrons on our electron carriers. We're just gonna basically put those electrons into the chain. What is the chain? It's a bunch of cytochromes, that's just what we call them, little proteins lined up along the membrane. And essentially, they allow the electrons to move through, and then the little proteins get excited and they do their little thing or whatever by the electrons moving through. That electron's got a negative charge, so it's going to help just pop some positive hydrogen out across the way. We're making them because it went NAD and NADH. Now we're taking the H off. We've taken the electron off and put it through this crap. The H plus is chilling. So we're going to pump the H plus out as a result of moving that negative across. Negative, 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 positive, 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 okay? Positive out, negative across. Now we're creating a gradient, a buildup of that positive on the outside of the cell. And as a result of that buildup, now we know about diffusion. We know everything wants to be equal on both sides. That's physics. That's called entropy. That's what that is. It always wants to even out. So do the protons. It's positive out there now. It's kind of negative, respectively, in the cell. So they want to come back in. The only way they can come in is through ATP synthase. So it's a little bit of a channel, but also an enzyme, clearly, ACE. And guess what it's going to do? Synthesize ATP. It's in the name. So as those positive molecules move through a protein, we know how that can affect the shape of a protein, and it does. That sort of activates ATP synthase to do its job. Pop on a phosphate onto ADP and turn it into ATP. Because we moved all these electrons through, and it was basically powered by moving the electrons, we call this oxidative phosphorylation, moving electrons, oxidation and reduction, right? Okay. So that's how we get our ATP. Nice, we got that far. Whenever we created all that positive on the outside, it's sort of like, because it wants to come in, it really wants to get in, it creates its own little force. We call it the proton motive force. It motivates the protons to want to come back in, creates a force that way. That's why it's called that. That's the word for it. That's the buildup of the protons. Okay, and it's an important one to know in order to understand all of this. Proton motive force, and the means by which we got the protons out by shuttling those electrons across and pushing the protons out. That's called chemiosmosis. All right, so we got through this part. Aerobic respiration. Now we've got our ATP made, we shuttled electrons across. Now we have electrons just chilling. They don't go away. They're still existing in matter. Physics says they don't just disappear, right? Unless it's like dark matter. So hopefully it's not there. We have our electrons chilling as a waste product, sort of. We've brought all these positives back in. If we let the positives build back up, we can't have the gradient. We can't have the difference between the outside and the inside. So we need to start getting rid of those pretty quickly. So the H pluses plus the electrons will then essentially make just H's that we can pop on with half of an oxygen, the O2, to make water. See, that's where we get rid of our waste products. That is the reason why you breathe in oxygen. It's the only reason why you breathe in oxygen. So we breathe in oxygen at this point here, at the very end as our final electron acceptor. During the Krebs cycle, a little bit before it, when we're breaking all these bonds and kicking off carbons as carbon dioxide, that's why you breathe out carbon dioxide. Notice that the oxygen, geez, freaking gnats. Notice that the oxygen in carbon dioxide is a different oxygen than the oxygen you breathed in, okay? So that exchange of those like waste and the oxygen coming in your red blood cells are not exchange of the same oxygens or something coming back out or anything. It's not the same. It came from food in the carbon dioxide. It came from air in the oxygen. So yeah, cool. So this happens in our cells and eukaryotes in the mitochondria. The mitochondria have these folds in the membranes in there that are called cristae. So that's where all these cytochromes are going to line up and all this is going to happen. All right. In bacteria, this happens on their inner membrane. We also call that the cytoplasmic membrane. It's the same thing. Okay. They don't have organelles. So everything pretty much takes place along that. All right. Can you believe I'm moving on from that? You would think I would be going on and on about more things you need to know, but it turns out you don't need to know more than that. All right, photosynthesis. Photons, light particles, essentially, yeah? They're going to excite pigments like chlorophyll in a chloroplast and create that same photon gradient that's similar to the proton motive force, but using the energy and excitation from photons, not electrons. Okay? So they get excited, we make the gradient, they come back in, we make some ATP. But it only makes a little bit of ATP, it's not super efficient, but it makes enough that in the dark reactions, these are reactions that don't need light. Light has to have light, dark just doesn't have to have it. It can happen in the light still though. Right, so we're going to use that little bit of ATP that we made to fix that carbon dioxide, which interestingly enough, the carbon dioxide that we exhale, we broke off of like glucose essentially, yeah? Now the plants are going to take that carbon dioxide that we exhaled and turn it into glucose. Here, they're going to reform those bonds and make it the organic molecule glucose. Why are they making glucose? Well, they can use it for things to build cells and all the parts in their cells and all that, cool. But the big daddy for them, again, same as us, plants have mitochondria, they need energy too. So they are going to go through cellular respiration just like us. Yes, they do the same thing to get their energy. So that's why they need glucose. They need energy. That's the real way they're going to make the energy. Look at that. So that was that. I'm going to see. Okay, now we have the choice between 9, 13, or 14. Any preference? 13 and 14 aren't that bad. I feel like they're vocab. I feel like 9 kind of is too. Yeah. Is there anything from the first exam that you guys want me to go over? Not really? Again, just going back and restudying. Okay. Do you guys have any questions about anything? Yes. No, I won't make you guys know any of the stop codons or the amino acids. None of that. Just know that they are amino acids and there is a stop codon. Okay. So the concepts, that's more important to me because I know that's not useful for you guys to know like a stop codon when you guys go into nursing. But if you guys end up working in some sort of field that involves genetics, I need you to have a basic concept of it, what's going on, which is way more common than you think, honestly, nowadays. But anyways, a lot of treatment is heading in that direction. Right, any other questions about the test, about the content? Yes, the lecture test is on Tuesday, right? After the lecture test, we will have the lab review. In here, you don't want to come? Don't come. It's not required, okay? I will be here for those that do want to come and do the lab review on Tuesday after the exam. So cool, because I posted the little Kahoot thing. It's that we're doing that. So really the benefit for it is if you want to come and talk to me about the stuff that's up there or ask questions about that or about maybe, I don't know, the nature of the questions themselves. I don't know. Yeah. What all of it could be on the test. So if you had to know it for the first exam, you have to know it for the second exam. So I mean, I would just go back and read, like I said, restudy. It will just be like relearning. You've seen it before. So reintroduce that back to your mind so that it's not different. What am I trying to say? A lot of these words we've been using since too. So a lot of the stuff that we learned in the first exam, keep in mind, guys, we've keep using those words and now you know them without even realizing that you know them, really. So that happened. You'll be surprised. So when you go back to look at the first chapters, I think that you'll find it a lot easier than you did the first time. I have somebody requesting for chapter 14. We have a little bit of time. We don't have to start lab until 11:30 and nobody's going anywhere. So I mean, like, right? We just do this. You guys want to keep trucking on this. And we're just, yeah. So we'll do 14. Sorry, we have a request. That's why I'm asking. I have a student that asked for it on here. What's up? You have a question? Anybody have any questions before? Okay. The epidemiology one. Highly vocab based, right? So that's what really I can say about this whole thing with epidemiology, or dealing with the population data, morbidity, mortality, prevalence, incidence. So essentially morbidity and incidence are going to be the same thing. I think I actually put the same definition there, FYI. But yeah, so just know the differences between them, be able to pick them out if you're given a choice in a multiple choice. Endemic, again, I feel like a lot of these endemic, pandemic, epidemic, sporadic, these are all going to be, know your vocab so you can compare and contrast. Those aren't too difficult. We've seen a lot of them. The R naught, that's going to be like if a person is sick with COVID, let's say, how many people are they likely to infect while they have COVID? So that's what the R naught is. R naught for COVID is somewhere around 3 to 5, depending on the strain or variant. So that's an example there. You're likely to infect 3 to 5 people if you have COVID. Case fatality rate is the frequency of deaths from a disease, frequency of deaths compared to people who get infected. So it is a percentage-based thing, and it is based only on if no treatment is given. Then specificity versus sensitivity. This is me watering it down. Specificity is determined by how many people test false positive, and sensitivity determined by how many test false negative. All right, so that's that. Then we talked about epidemiological studies. So these are probably new concepts to some people. We have cross-sectional studies where we have 2 factors, like how was the person exposed versus like what the disease symptoms were or something during this outbreak at one time, but we don't know which came first or anything, but it gives us a cross-section of the relation maybe between those 2 things in that outbreak. Case control, people are separated as positive or negative. Then we're going to ask them about things that we think are related to the exposures. Who was exposed to this and who was exposed to that and which ones got sick. Like if Jon Snow, from the whole story of it, were to come up and ask people, okay, you're sick. Which well did you drink from? You drank from well B, you're not sick. Which well did you drink from? You drank from well A, you're not sick. So it's probably well B that's making people sick, that sort of thing. So that is a case control study. A cohort study is going to be looking at uninfected people over time and just watching to see as they get infected, and then going and being like, okay, so you got infected, let's see the breakdown, where we think that you got this from, because you can see it from the start from that kind of perspective. The criteria that you need to meet in order to say that something is a cause of an outbreak is called Hill's criteria. And look at that, that's all you need to know from that, right? So hospitals and neglected diseases, hospitals, medical asepsis like washing your hands, gowning up and clean disinfecting surfaces, surgical asepsis, really working towards true sterility, no microbes at all in the area. Can't keep that up everywhere. And then the infection control officer that, you know, make sure that everybody's following those rules and like tries to figure out why you might be having an outbreak at a hospital if everybody's supposedly following the rules. Next, the neglected diseases in the United States. We're just, there they are. Listed. They're all, these are parasitic diseases, so they're either protozoan or helminth in nature. Okay, so that's basically 14. Like I said, this is a vocab test, if anything is. Yeah.
That's what I was thinking.
Yeah,
Maybe
13.
fine.
We got a couple minutes. We can keep going. We can go over 13 real quick. And we can come back to 9 if we want. Okay, so 13. 13 is, we start off talking about a little bit about the biome, right? So what is the biome? Basically the bacteria that live normally on your body. Could be different from mine. So it's just relating to you. Microbial antagonism is kind of the purpose that that biome does serve for us. So there's a bunch of them taken up. Again, the stadium seats. If the stadium seats are taken up, the bad guys can't come sit down. They can also make like deterrent, like chemicals, like toxins or whatever, to drive off bad guys as well. So microbial antagonist, like an antagonist in a story. So right, we know kind of the order of how you might get exposed. The idea now, the first exposure, and I guess this is like a little bit different, but it is the first exposure in utero. Okay, we think we're actually getting it in utero while you're growing, and more in the birth canal, more in the breast milk, then from caregivers, and then eventually from the environment that you're interacting with. It keeps building up and changing your like gut biome, and how it might break down sugars for you that you wouldn't be able to interact with, and a lot of other things. But yeah, you're never gonna stop growing your biome. You're never gonna be done with that. Anytime you have a new exposure to like a new person, like a new sexual partner or something like that, that could lead to your biome changing. All right, why is this making this noise? Oh, it's this door. Okay. Bacteria, fungi, yeast is fungi, by the way. Viruses and protozoa. So those four: bacteria, fungi, viruses, protozoa. Those types of microbes can be part of our biome. They're not helminths. Helminths are the worms. We don't have those naturally. Sometimes our own biome can betray us whenever we have opportunistic infections or moving the microbes from one place where they belonged to areas where they don't belong. Like we were saying with the urinary tract infections coming from bacteria from your own rectum area. They don't belong there. And of course, you get sick from one thing, that can make you more susceptible to getting sick from another. That's an opportunistic pathogen. Disease is any deviation from health. So pathogenicity is a term where we're talking about, causing disease. So you know that you're causing disease. You're starting to notice that you're not healthy. That's pathogenicity. Whereas virulence is like a measure of the degree of that, like how bad it is or not. Okay? So those are different terms. Pathogenicity, just having the disease and knowing that you have the disease. Virulence, the degree of how bad, the symptoms and everything like that are. So remember, signs versus symptoms. Signs can be measured. We call that objective. Symptoms, those are subjective. They can't really be measured. You just have to go with like the patient's word, like saying that they're in pain, for example, headaches, stuff like that. And how does infection progress? We have entry. You have to be introduced somehow, maybe through the skin, maybe through a mucous membrane. Those are the most common ways. Adhesion of the microbe to some surface so that it can start growing in that area. Evasion of the immune response, and even specifically phagocytosis, where our sentry cells are going around eating bad guys up. Like capsules help with that, right? That's one of the things that we learned about definitely that I would be aware of. It's going to come up over and over again. Then we have damage that can be caused during the course of the disease that often presents as the symptoms and signs for us. We talked about exotoxins versus endotoxin. Exotoxin, they make a toxin and put it outside of the cell, and it can cause damage or be poisonous to our cells. Endotoxin is part of the cell, right? It's only gram-negative bacteria that have that, and it's on their outer membrane. It's also called lipopolysaccharide or LPS. That's all the same. All right, and then we have to get out of the body so we can go infect the next victim, and that's how the disease will progress. as far as when you actually have the infection and how you start seeing it progress, not just how the microbe grows in you. Now the disease itself will have an incubation period. You got infected, but you don't know that you're infected. It's just starting to grow. Your body hasn't recognized it, everything like that. So flat, sort of like the lag phase in bacterial growth curve. Next, we have the prodromal phase. That's introduction of the first recognizable symptoms that will lead into the acute phase, which is increase of symptoms and leading to like the worst of the worst. So symptoms, you usually will plateau, right? And then as you are starting to recover or getting ready to die, you have convalescence, where we'll have a retreat of that symptoms and recovery, hopefully, right? That's the end. So anytime, we've seen this term before, but anytime that you have a pathogen that's going to hide out in your body, like chickenpox, like herpes viruses, and they're waiting to come out, so things are just right, that's called latency. And that can account for bacteria can do it too. That's just a term referring to that state, sort of. And then we have sequelae. That's a term that's talking about effects of an illness that will last even after you've cleared it, like people who get paralyzed permanently from polio. And then zoonoses are diseases that can be primarily maintained in animal population, but we can get them from them, like rabies. That's an obvious one. That's that. All right. Anybody want me to go over chapter 9 or anything like specific from the early chapters that you guys have questions about? All right, we good then? Well, we got this mostly done. I hope that was helpful for you guys. Of course, when you're studying, if you have questions about something you're confused, or even if you want to know, you're looking through something that's got all these steps and you just are like, do I need to know all this? I don't want to waste my time studying it. Then you can email me and at least try to ask. It doesn't hurt to ask. So at least try to ask, and I'll hopefully be able to give you an answer about that. But yeah, I don't need you to know details beyond this. You'll notice that I went through the Krebs cycle and I didn't talk about any organic molecules. I don't want you to know that. It's useless. I want you to know what the purpose of it is. Yeah. So that's the idea there. We've got the picture of it. Yeah, I know when I learned the Krebs cycle, I took biochemistry like proper, like it 3000 level. And that was my major in college at OSU. I had to learn all this stuff, all that. I had to be able to like explain what was happening in each step too. Oh my God. I'm not gonna do that to you guys. So okay, if there's no more questions for that, then I'm gonna go ahead and stop recording for this review and upload this for you guys and for everybody else to look at eventually when it comes back to me. And we'll do lab. Cool. I'm glad we were able to do it. Here's my cursor.